So we jump to the Neil Desai's presentation. As you know, he's the father of the NAP technology. This is very well known for the clinical development of Abrexan. He's now founder and executive chairman of Aadi Bioscience in California, in USA, which is focused on gene modulation using peptide-based delivery of nucleotide therapeutics too. More than 100 issue paper, more than 40 peer-review journals. So, Neil, thank you very much for joining us, and please go ahead with the presentation.
Yeah. Hi, can you hear me okay, Seema?
Yes, sure. Very well.
Okay, and is the slides up on the screen? I can't tell.
Yes. Yeah, they are.
Okay, very good. Well, thanks very much for the opportunity to present at ClinAm. Of course, I'm sorry I'm not there in person, but due to some issues, I could not make the travel. Today, I want to talk about something a little bit different. I think you know about the NAP technology, but we want to talk about how we can think about registration of these drugs in a different way in a tumor agnostic setting. And that's what I will discuss. So I'll talk about FIARO, which is our drug. It's NAP sirolimus. I think all of you are familiar with the NAP technology from quite some time. You're familiar with Abrexan, which is the first drug that we developed using the albumin platform. And so this is the next one, which is sirolimus, and there's an mTOR inhibitor. And we got this drug approved in the end of November of 2021. And I want to speak to you about that a little bit. So it's again albumin bound to sirolimus and then delivered in the NAB platform. And the focus here is cancers that are mTOR dependent. Now, I think most people know about the mTOR pathway and its importance in cancer and actually many other diseases as well. It has very important pleiotropic effects and is connected to many other sort of RAS pathways, LKB1, PDK, and other things. And a lot of these signaling flows through mTOR, and which is why mTOR is very central as a target if you can hit it effectively. So the prior drugs with mTOR, which include everolimus, sirolimus, etc., have many problems because of poor uptake, not effective targeting of mTOR, incomplete inhibition of mTOR. But with this albumin binding, we're able to get very high drug levels into the tumor, and therefore we're able to suppress a target very effectively. And that's the concept here, is that then we go after mTOR-driven tumors and then have an effect. So the first one we went after from a registration path is a tumor which is a very rare form of sarcoma, which we found to be driven by mTOR more than any other pathways. And so this is where we looked at this carefully, sarcoma, and it's called PEComa. And a lot of the driving factor in these tumors is a mutation in TSC1 and TSC2 genes, which are just upstream of mTOR. And this actually ends up activating mTOR. So there was never previously a clinical trial in this advanced malignant PEComa. And therefore, the estimated survival was only about 12 to 16 months. And so we took this concept to the FDA and said, look, here's a tumor that seems to be driven by mTOR activation, and our mTOR inhibitor seems to target the tumors pretty effectively. And can we use this as a registration path for this drug? And indeed, we were able to agree with the FDA that because this was a very rare tumor and there was no prior clinical trials, that if we achieved a 30% response rate in about 30 patients or so, because this is a very rare tumor, that would be sufficient for approval of this drug. And so we conducted this trial, and this trial the name of the trial is called AMPECT, and this is our registration trial, which we conducted a few years ago, and the results. So the endpoint for the trial was overall response rate and for full approval. And of course, duration of response is an important factor because you want to make sure that if you get a response, that the response is not very short-lived And so you need benefit to the patient, and so you should have a sufficient time of response. So here's the waterfall plot showing tumor shrinkage for all of these patients. And you can see that for a sarcoma, it is quite rare to see an outcome like this, where you have almost 40% of the patients responding really well to the drug. And in fact, we had a few patients who had a complete response to the drug as well. What's particularly striking was that the overall response rate was about 40%, but what was very impressive was that the duration of response, the median duration was almost 40 months. So over 3 years of duration in a median measurement. And so that was pretty striking, and this was sufficient to get the drug approved. So in November of 2021, this drug was approved. FiArrow is the brand name, and today it is marketed by our company, Aadi Bioscience. Now, within this study, we were very interested in the genetic makeup of these tumors. And so we had done next generation sequencing, NGS, and genomic analysis for these patients. And so the next slide, I'm going to show you the same outcome, but in terms of the mutational status. And so here you see the same waterfall plot and response, but now grouped based on the mutational status of TSC1 or TSC2 genes, which are just upstream of mTOR. And you see that majority of the responders on the right-hand side of this graph happen to be the ones with either TSC2 mutation or TSC1 mutation. So it's clearly telling you that these mutations are important drivers in this disease of PEComa. And so if we look at the response rate for the patients who have NGS information, that response rate is even higher, is 64%. So 64% of all patients who had the mutation responded. And if you look at, in particular, the TSC2 mutation, that number is almost 90%. So it's a very impressive response and very good proof that these are important mutational drivers for the disease. So where does this lead us? Now, TSC1 and TSC2 is very important genes. They're tumor suppressors, and I'll speak a little bit more about that. But this is what got us thinking about a tumor agnostic strategy. And just to give you an idea of what that means, for those of you who are not familiar, so tumor agnostic or tumor tissue agnostic strategies are now becoming a new regulatory path. So with the evolution of precision oncology and genomic analysis, a lot of patients have the DNA analysis on the genomic sequencing on their tumors, and we can rely on this information to find what is mutated in their tumors. So rather than the conventional treatment and classifying tumors by anatomic origin, so say lung cancer, breast cancer, etc., we want to identify the tumors as driven by their genetic alterations. So the regulatory agencies have recently developed guidance and granted pan-tumor approvals across a variety of tumor types just based on the genetic status. So there's a tissue agnostic strategy available for tumors and drugs that are targeted to specific mutations. So to give you the idea of, since 2017, a few drugs have been approved in this setting. So Keytruda, of course, is the anti-PD-1 drug, and then the NTRK inhibitors, Victravi and Rozlytrek, and then later again, Keytruda for a TMB high in solid tumors. There's another anti-PD-1 which is also approved, Jemperli, and then a combination of BRAF and MEK inhibitors in V600E mutations. And then most recently was Retevmo, which is a RET fusion inhibitor. So these are drugs that have all been recently approved using the tumor agnostic approval. And all it takes is a single arm trial. So there's no comparator trial. This is important to note because you could not do comparator trials for all of these different tumor types that you treat. And they approved with a response rate endpoint and with a duration of response as major efficacy outcome measures. So this is also very important because it gets you an accelerated approval and you don't have to do randomized studies for the initial approval. So we use this strategy for TSC1 and TSC2 alteration. So this is the same mTOR pathway, and the TSC1 and TSC2 gene is highlighted, and this is a tumor suppressor. So a loss of function of the TSC1 or TSC2, which works as a complex, results in mTOR activation downstream. And this is where our drug FIARO comes in and can effectively inhibit the mTOR and therefore get results in this tumor. And so if you look at the analysis of what patients or what tumor types have TSC1 or TSC2 mutations, it's approximately 1 to 2%, roughly maybe 2% across all cancers. And we've done this detailed analysis, and you can see here the patient numbers based on the left side, either TSC1 alterations in blue and TSC2 alterations in orange, and the top histologies. So this represents US patients, new patients every year. So the top 5 in each category, there's over 1,700 patients in bladder cancer, there's over 1,000 patients in non-small cell lung cancer, almost 1,000 patients in endometrial, liver cancer, etc., depending on which mutation you look at. So there's a lot of patients who have these mutations, and they could be driver mutations for their tumors. On the right side, as a comparison, is the analysis of the same set of genes, but looking at all the other mutations that are targetable and that have drugs approved for them, or could have drugs approved for them. And if you look at TSC1 and TSC2 together in the green bar, you see that the numbers compare very well. It's in the top 6 of all mutations in this setting of targetable mutation. So this is a healthy number of patients every year that get these and have these mutations. So we looked at some analysis of other patients that we had done, and this is in our expanded access program. We look at PEComa patients who had failed other mTOR inhibition treatment. And so they've all progressed on other mTOR inhibitors like everolimus, sirolimus, or temsirolimus, and then come on to treatment with FIARO or nab-sirolimus. And this is the outcome. You see the waterfall plot here for those. So we had about 25% response rate in these patients. And if we looked at the genomic analysis, we found that all the responders had either TSC1 or TSC2 mutations. So again, important driver of the disease is the mutational in TSC1 and TSC2. We also looked at other types of tumors, so non-PEComa, and this included ovarian cancer, included other types of sarcomas like angiosarcoma, included endometrial cancer. And all of these patients had TSC1 or TSC2 mutations, and when treated with nab-sirolimus, we saw, despite the fact that they had failed 2, 3, 4, or 5 other prior therapies, they responded very well to the drug. And so this became a good rationale for going after TSC1 and TSC2 mutations. And I'll just, so looking at all of these different experiences with different patients, we have about 31 patients treated with TSC1 and TSC2 alterations, and the response rate in that population is about 58%, which is very encouraging, especially because a lot of these patients have failed multiple prior therapies. So this is a design of the ongoing registration trial, which is a tumor agnostic trial for patients with TSC1 and TSC2 inactivating mutations or alterations. The trial is called Precision 1. It has 2 arms. Patients, arm A is TSC1 patients, and there's about 60 patients for that. And then arm B is TSC2 patients, and another 60 patients for that. Again, the endpoint here for potential approval is response rate. And of course, duration of response is very important, as I mentioned before, because we want to make sure that patients who respond continue to respond and not, not progress in a few months' time. So that is the ongoing trial, and we hope to report that next year in the full trial in 2024 with a total of 120 patients accrued. But later this year, we will have an interim analysis on the first 40 patients. So now looking at the pipeline for Aadi Bioscience with FiArrow. So the drug has been approved in malignant PEComa and is being commercialized as we speak. We have the Precision-1 trial, which is ongoing right now for the TSC1 and TSC2. We're about to start a new trial in endometrial cancer, where mTOR inhibition is very important. So that's in combination with letrozole. We have a neuroendocrine tumor trial, which is also about to start soon. And we have a combination trial with the KRAS inhibitor with Mirati that's ongoing right now. And there's a very good synergy that we've seen in preclinical setting between KRAS inhibition and mTOR inhibition. So with that, I'll stop, and thanks for your attention.